sure some of you know, or maybe most of you, I work for Nationwide Children's Hospital. I'm on the foundation side, raising money, and so it's always fun for me to be able to introduce someone who works at the hospital. Senior Director of the Genome Informatics Group in the Institute for Genomic Medicine at Nationwide Children's Hospital. So Genomics, you got that? Please help me welcome Dr. Peter White.
Thank you very much for this opportunity to come speak to you today about some of the work that we're doing at Children's in this area that we call genomics. So I'll explain what that is for you as I go through my talk, and then we'll have time for some questions afterwards. But the field of genomics really began with the Human Genome Project, which was an international effort that started in around 1990. It wasn't completed until 2006. But this was an effort that was to sequence the first human genome, to actually get what is the sequence of our DNA, the A, C's, G's, and T's that make up our DNA. And that took about 15 years to complete, and they estimated the cost to be about $3 billion. So the human genome is made up of 3 billion A, C's, G's, and T's, so it's about $1 for each one was the final cost. And this is what it would look like where you'd print it out. This is housed in the Wellcome Collection in London. It's about 110 volumes, each with thousands of pages, with just the characters on each page of A, C, G, and T. And the gentleman in the middle there is Jim Watson, as in Watson and Crick, one of the scientists that discovered the structure of DNA in around, I think, 1953. But with completion of the genome, the Human Genome Project, we entered what you probably haven't heard of this, but we're actually in the post-genomic era. We've sequenced the genome. What are we actually going to do with it now we've got this genome sequence? And so this gave birth to this new field that's called genomics. And genomics is defined as a discipline of genetics that applies molecular biology, DNA sequencing, and bioinformatics, which is the analysis component, to sequence, assemble, and analyze the function and structure of genomes. Now, the root of the word genomics is from the ancient Greek genesis, which means origin. So when we're looking at a patient's genome, what we're actually doing is we're looking at how is it that the DNA that they've inherited from their parents, how is it that that DNA is contributing to the origins of this particular disease that they have. Now, at $3 billion, we wouldn't be sequencing many patients, but fortunately, over the last 20 years, the cost of genome sequencing has fallen. Back in 2001, it would cost about $100 million to sequence a genome using a technology that was available at the time. But it wasn't until 2007 that the pricing really began to fall with the invention of a new type of sequencing technology that's called next-generation sequencing. And we got our first next-generation sequencer around 2010 at Children's. And this enabled us to start sequencing human genomes for about, back then when we got this first machine, it was about $20,000 to do a genome and took around a month to do the sequencing. So that time frame had shortened dramatically, but it's continued to shorten over the last decade. And today, like in 2015, thanks to a gift from the Nationwide Foundation, we're able to update our technology, and I'll show you this new instrument on the next slide. But our cost to sequence a human genome today is around $2,000, and you'll hear a figure out there that sometimes as low as $1,000. There are much more expensive instruments than we have that can drive the cost down further. The goal is one day to get it down to about $100 per genome. At that point, sequencing a genome could become part of like routine screening at birth. But this is the technology that enables us to sequence a genome today, this next generation sequencer. It actually occurs on the surface of a glass slide. That slide in the middle there is about a 3-inch by 1-inch glass slide that contains many billions of these micro wells where a sequencing reaction occurs in parallel. So there's about 1.5 million of these reactions occurring on each millimeter squared in the surface of this slide. So today we can sequence 12 human genomes in about 4 days. And again, this technology has evolved just recently. The Illumina announced their newer machine that takes that 36 human genomes every 40 hours. So we're able to sequence genomes much faster and much cheaper. And it would be great, though, if these sequencers actually produced output that looked like that volume, the volumes that are housed there in the library, but they don't. How we sequence a genome is our chromosomes are very large. The largest is around 250 million bases long. We have to chop those down into very small fragments to do the sequencing of about 300 nucleotides each fragment. So it's a bit like taking that entire volume and running it through a high-capacity shredder. We shear everything down to 300 bases, perform all those billions and billions of sequencing reactions, and then have to put it back together. And that's where my group came in, was to sort of look at the analysis challenge of reassembling this mess. But not only are we reassembling it, we have to do it very accurately, because when we're looking at a patient with a disease, we're looking for that one page that contains a typo. And I'll zoom in for you, because this page contains a C that's changed to a T. And this change, this single base pair change, is responsible for a disease, and this could be responsible for a disease in any given patient, as it results in a change in a protein that has an essential function in the body. So for us, when we first got this machine, we ran into a problem very quickly. It was taking us about 2 weeks to do the data analysis, and a machine that could generate data every 12 genomes every 4 days, and we could do analysis of one every 2 weeks. We rapidly ended up with a massive backlog of data. So we really looked at this problem of the analysis, and we actually came up with a method that I'm not going to go into any details of the computer science behind it, but it's a method that we call Churchill. I don't know why we picked that name, but it came up with that name. But essentially what it does is it parallelizes the analysis across many computers simultaneously, and we're able to take that 2-week window down to 2 hours to go from the raw data to here's the genetic variant that could be causing disease. But because we're using this diagnostically, we want to use this in a clinical setting. For us, it just wasn't about the computer science research that went into this. It was also about maintaining something that would work with high accuracy and be highly sensitive. And the National Institutes of Standards and Technology actually released what they called a reference genome for people to be able to say, how well does their sequencing technology work? How well does their analysis technology work? And what we did is we compared Churchill against analysis methods from the Broad Institute in Boston, which is the GATKQ results up there, and then HugeSeq came from Stanford University, and then SpeedSeq was from Washington University in St. Louis. And what we found with Churchill was across the board, we had the highest rates of detection, the lowest error rates, and best sensitivity. And also the method's 100% reproducible. So every time we run this analysis, we get the same results, which again in a clinical setting is important, because we don't want something fast that isn't going to be accurate. So having solved that problem, what are we doing with it? What are we doing with the genome sequences and the analyzed data? Well, these are my twin daughters, Samantha and Charlotte. Samantha is Thing 1 on the left. Yep. Yes, Samantha's Thing 1 and Charlotte's Thing 2. And they're actually genetically identical, and I really struggle to tell them apart. My wife has no problems whatsoever, but I struggle. But what you can't see from looking at them is that Samantha was born with a congenital heart defect, abbreviating that as CHD, but her sister Charlotte was born with a healthy heart. This is actually Samantha when she was 1 day old. She was born here in Columbus at Riverside, and very quickly they realized she was having trouble, and she was rushed into the neonatal intensive care unit there, which is any parent, not how you want this for what you're expecting when you're having a baby. But she received wonderful care there and is actually doing fine. This is her a cardiology clinic visit last year. She's looking quite concerned. She's having an EKG, so she's got about 19 wires on her. I'm actually, if you're close to the front, you can see I'm actually beaming, because I'm like, this is going to be great for every talk I give. I'll be able to show this slide. My wife's like, why do you want me to take a photo? I was like, just take it. And then this is us leaving the hospital, and she's actually, her and her sister are cuddling one of the animal friends around the hospital. But congenital heart defects are the most common type of birth defect we see. About 1 in 100 kids are born with some form of heart defect. And we know that genetics is playing a role in this disease because we often see it occurring in families. But so far we've identified very few genes. We really don't understand much about this disease. And so what myself and my colleagues, Kim McBride, who's a geneticist, and Vidhu Garg, who's a cardiologist, we've actually been funded by the National Institutes of Health for the last 5 years to look at families and use this next generation sequencing technology in families to see if we can find genes that are uncommon in patients that have a heart defect. We've actually been fortunate enough to sequence over 700 different cases, and this is a photograph of our very first family. They have 6 children, and from a genetics perspective, this is a great family to study because it allows us to look at the 3 children that are shaded, all had a ventricular septal defect, which is a hole in the wall that divides the 2 ventricular chambers. This had to be corrected surgically after birth. But 3 of their children were healthy. So in sequencing the whole family, we could look and say, what is it that the children with the disease have in their genome that the kids that don't have the disease that don't have? And we're able to find a new gene that we believe plays a role in early development of the heart as a result of this sequencing. Another area that we're really interested in is rare diseases. Now, rare diseases are exactly that. They're not very common, but together as a group of diseases, they are more common than you would expect. Just in 2015, Boston Children's actually had a competition that they called the Clarity Challenge. And what this was is they had 5 families for which they knew they had some sort of disease, but there was no diagnosis. And they performed genome sequencing on these families and then asked the international community to look at this genome sequence. They'd looked at it, they couldn't work out what was going on. And so they were like, let's turn this into a competition. Let's crowdsource the genome data. And so these 5 families all volunteered to have their data shared around the world. And Children's took part along with 26 other teams. And we actually won the competition. We were able to find a potential answer for 4 out of the 5 families. And we're also cited for not just our analysis approach that we took to it, but we were the only team to think about communicating this data back to the families. We had our genetic counselors write letters to the family explaining what the analysis was and what we found in language that would be understandable. So that actually gave birth to a new program at Children's, which has been our own rare diseases program. To date, we've had about 36 families that we've been able to enroll into this research study, trying to identify what could be causing the disease in their particular in their children. So how do we see this moving forward? Where do we see this genomic technology going as we look to the future? Well, if you were reading the Columbus Dispatch last May, they published this article announcing that Children's had landed 2 of the top genomic scientists, and these are Rick Wilson and Elaine Mardis. And these were like 2 of the, in my field, I was like, these are rock stars. It would be amazing if we got them. And they actually decided to come to Children's, and they started just this fall. And we formed a new institute for genomic medicine. And the vision for this institute is we want to see we believe that genomics could really transform medicine. And so we have a mission to see how, if we can introduce genomics into the everyday practice of pediatric healthcare. And there's a number of areas that we're going to focus on, but one is definitely in the rapid diagnosis of genetic disease. So this is an example of one of those rare disease families. This was a family that came to Children's just over 5 years ago. Their newborn daughter was critically ill, and shortly after arrival went into a coma in our emergency department. They were able to treat her and stabilize her, but as her blood pH was dangerously low, and what happened as a result of that initial presentation, she suffered severe damage to her central nervous system, and at the age of 5 is unable to walk, talk, or even sit well. The family, having been treated though, the family then underwent multiple genetic tests to identify what is it that caused this disease. And after years of testing, they were unable to find any cause. And this is when they were enrolled into our rare diseases program. And what we found in performing next-generation sequencing on the child's mum and dad, and the affected child shown by the red circle there, and her unaffected brother, we found that she'd inherited a bad copy of this gene called CACT. One bad copy came from her dad, and a different bad copy came from her mum. And as a result of that, she had no functional copy of this gene. And this led to this disease. We were able to diagnose the disease for the family and give them a diagnosis. And the family was also able to receive genetic counseling in terms of their risk moving forward with additional pregnancies. But the disease was called carnitine acetyl carnitine carrier deficiency. And this is something that could have been easily treated with oral drops had the diagnosis been made when the child was born. So it was a metabolite she's missing, could have been very easily replaced. So our vision is that if we have children in our NICU that we suspect that have a metabolic disorder, that we're going to introduce this testing so we can get to this diagnosis rapidly, because a lot of these genetic diseases, their harmful outcomes can be prevented if treatment can be tailored early on. And we actually this January launched our first, so when we first did this was done in a research setting. This had to be moved into a clinical setting, into a clinical diagnostic laboratory. And we started doing this in January clinically for the first time. And I think we're up to our 50th case clinically, 50th order clinically just this week. So it's been rapidly adopted throughout the hospital. Now, the other area that we're very interested in is in cancer genomics. Dr. Wilson and Dr. Mardis have been leaders in the fields of cancer research. And this is a story that sort of highlights what's possible with this technology in regard to cancer. The gentleman in the photograph there is a doctor called Lucas Wortman. He's a researcher from St. Louis. He actually worked with Dr. Mardis and Dr. Wilson in their lab. And he was diagnosed with leukemia at the age of 25. He was treated and went into remission. But then at the age of 30, he had a relapse and had to undergo a stem cell transplant from his younger brother. Then 3 years later, he had a second relapse, and this time he was seriously ill. It was involving his central nervous system, and he was running out of options. And they decided at the time they would sequence the genome of his cancer cells. So they isolated the cancer cells, they sequenced the DNA in those cells, and they also looked at expression of the genes. They looked at the RNA as well, like to see how highly genes were turned on or off in the tumor cells. And what they found, Malachi Griffith was a postdoc in the lab working on this project, and what he observed when he compared Lucas's cells to normal donors was he had extremely high expression, it's shown in that red circle at the top left, extremely high expression of a gene called FLT3, which was actually a target for a drug called sunitinib. This was used as I can't remember the type of cancer it's used to treat, but it hadn't been used in leukemia previously. But they realized it could potentially be a target for tailoring therapy for Dr. Wortman. After 12 days of treatment with sunitinib, he went into complete clinical remission and was able to receive a stem cell transplant. Now, there's two outcomes for this story. One, Dr. Wortman's alive and doing well, like 4 or 5 years later now. And secondly, we discovered FLT3 was a new, the discovery of FLT3 was previously unknown to play a role in leukemia. So it's now been added to the panel for screening, so people are screened for this expression of this gene because they know this is a successful way to treat the disease. So that's part of our vision for this technology with our kids and children, is that we can apply similar approaches. And we've just had our first case this week. Maybe if you'd given me another week or two, I'd have been able to present some data on that. But we had a newborn child that actually had a brain tumor, and it's very unusual to have a newborn with a brain tumor. So we were able to, he had surgery to remove the tumor, and we've been able to sequence that DNA. And what we're doing, we're looking for something. Is there any signature there that could tailor treatment for this kid? So more on that, maybe if I come in a year's time, I can give you an update on that story. So I'll finish up by just sharing with you some of our short-term goals. That's a photograph of the Institute for Genomic Medicine. It's right across from the hospital on Parsons. My lab moved over there in the fall. The rest of the second floor is being completed this summer, and our clinical lab will also move over. It's currently in the basement of the hospital, and it will move across the street there. June or July time is the plan for that. We really are expanding our sequencing efforts, moving into the neonatal intensive care unit, and also our expansion in working with the hem-onc department, and we want to sequence more patients with cancer. Then the undiagnosed disease research. This is something that I've felt very passionately about doing. It often doesn't result in a treatment, but it's very important to give families an answer. It's very painful when you're in this situation, searching for an answer of what's wrong with my kid. So this has been something that's been tremendously rewarding for us in terms of the research we've been able to do, but also for the families that we're able to give answers to. And then finally, in the area of data analytics, as I said, the genome's big, and as we collect more and more data, we have to optimize ways that we can analyze it and looking for patterns there. And that leads into our long-term goal, which is we believe that genomics is going to underpin many of the different clinical practices, from everything from cancer, infectious diseases, heart, gastroenterology, neonatology, and neurosciences. And we're envisioning a future where a mill of blood is collected from each of these patients. Their DNA is sequenced. The data from that is stored in one of these in an optimized database that's been really tailored to look at the genomic signature and allow researchers to start looking at the data and say, are there any markers in this patient's DNA that could be used to help improve treatment in terms of rapid diagnostics, predictive analytics, or optimizing treatment, with the end goal being that this personalized management plan will result in better outcomes for our kids. So with that, I'll finish up, and I do want to say thank you. Thank you to you, the members of the Ohio community. Without your support, the research that I'm describing here and lots of the other research that you haven't heard about that's going on at Children's wouldn't be possible. And also the Nationwide Foundation and the National Institutes of Health have helped support some of the research that I've shown you. But happy to take any questions.
Share with us what kind of funding you're getting from the National Institute of Health versus private sources. I mean, what's your annual budget?
So the funding with the National Institutes of Health has been declining rapidly over the last 10 years. So more, I don't know the exact percentages. I think about is it a third? Would you say is coming from the government funding, and 2/3 is coming from private funding.
And it seems like funding seems to be coming from Asian sources.
So the question was, there's funding coming from Asian sources, like in countries like Hong Kong, where genome sequencing is done more affordably. And could I comment on that? So I think the largest Asian genome sequencing I know of is the Beijing Genomics Institute. They had an initial focus of not just looking at human genomes, but also looking at, they were part of the Rice Genome Sequencing Project and other sequencing projects. I'm not sure. We've looked at some of the possibly sending samples there for sequencing, but it's around that same price point that we have doing it in-house. So. I'm not sure how they've been getting funding to sequence as many genomes as they have done, but there's definitely been a number of efforts internationally to sequence populations, entire populations. Like the United Kingdom has a project to sequence 100,000 genomes as part of the National Health Service. And then we have here in the States, there's the Precision Medicine Initiative, I think, is to sequence a million genomes. I think that will be one of the largest projects, and that's just beginning at the moment. Yep. So the question was, what are the potential uses and misuses of this technology from insurance companies? I have to be careful what I say about this, because a lot of our funding comes from an insurance company, Nationwide Insurance, has been, but no. So one of the things for health insurance, there is legislation in place that prevents people, in terms of the being discriminated against, I think, for both employment and health insurance purposes. There's legislation in place that prevents that. Where it could play a role is in life insurance, that if they are able to link your genome signature back to a life expectancy, then I think it could definitely be used there, that there could be valuable data to gain to predict, okay, this person should have a higher premium based on their genome sequence. That's not happening today, but I could see a future where that is happening. But good question. Craig Hoyt.
My daughter has a chromosomal challenge from birth, and I'm wondering how genomics whether it's being used to research chromosomal abnormalities.
So the question was about chromosomal abnormalities. How is genomic technology being used there? And absolutely, it's being used in that area. So what I described is looking for very small mutations, like that single base pair change, But what we're able to do with this technology is get a much more accurate picture of the entire genomic landscape. So this is looking for chromosomal abnormalities that are normally larger changes in the underlying DNA, either a large deletion of a piece of a chromosome or not. And the current technology to look at that is limited to about 100,000 base pairs is the sort of limit of detection. But using this next generation sequencing technology, we can actually look and see much finer detail if there's smaller genetic abnormalities that are there. And we can also detect rearrangements. So some of the cancer cases that we've been looking at, we actually see the DNA is rearranged. And this rearrangement introduces, like when you look at it in terms of a copy number of the DNA, everything looks normal. But this rearrangement actually brings two genes together that shouldn't be together. And that can be a driver in cancer. So this technology is being applied to look not just at that very fine level, but also at a more higher view of the DNA.
To drive sequencing costs down to maybe $100. And in the report that I was listening to, that I was discussing, they were talking about newborns are estimated to have perhaps as high as a 3%, I want to say genetic imperfection. I'm not sure what the right word would be. Do you agree with that?
The 3% figure would be very high. For when we're looking at like what we call a trio, when we're looking at the sequence data from a mom, a dad, and a child, we can look and see how many of the changes in this child are new errors. And typically out of those 3 billion, we see about 60 changes that are what we call de novo, they're spontaneous changes. So 60 out of 3 billion is much, much less than 3%. So that figure seems high for it to be that high. But it does happen, and that is a common mechanism by which disease comes about, can be those spontaneous changes that happen. But thank you.
And we have a guest that has one question.
Guest with one question, and this will be the last question.
Ask a question. Because you mentioned the neurological aspect, are there any teams that have already started to do research when it comes to Alzheimer's and the possibility of Alzheimer's having genetic?
It's about the neurological implications of genomic research, and especially in diseases like Alzheimer's. So Alzheimer's is a disease for which they've found some of the, like not all Alzheimer's, but a large amount of it can be attributed to changes in certain genes. So there are known genetic causes of that. But in terms of more broader neurological research, we've been very involved in looking at kids with autism spectrum disorders, trying to understand, is there any genetic basis to that disease? And so far, we're not finding strong evidence for genetics in the disease. Maybe 5 or 10% of cases, we think we've found something that looks genetic, but the vast majority, we can't find anything. But in our neurology clinic, they've been, of the different clinics that I showed in that last slide, neurology has been the biggest adopter of this technology. It's able to be applied to multiple conditions from periodic fever to other neurological conditions. They're able to use, they're starting to use it more and more. So it's an active, again, there's so much of the genome that we just don't understand, but we're finding more and more. And as we find more, then its applications start to get broader. But good question. Thank you.
Dr. White, thank you. Great presentation. Thank you. And to all our students and to our award winners, thank you very much on behalf of Columbus Rotary. Good luck in selecting your careers, and this meeting is adjourned.